if you can crystallize a molecule, you can give applications differently. The crystal chemistry, the extraction, for example, was known in the 18th century. 1747 में ही जो है, they knew कि extract करके crystal को कैसे बना सकते हैं. How you freeze it and convert something into crystal, right? Crystal की applications we know, sugar खाते हैं. That's a crystallography. जो crystal उसको मतलब crystallography in the sense to converting into a crystal, जो solvent है उसको लेके. So that technique became more and more. Sucrose जो लिखा है, I wrote the crystalline sucrose, वो was shown in 1747. How you can take the sugar and it becomes actually a crystal, right? So that when that technique became more and more, solvent extraction became more and more, knowledge came out, and now the same. Now think of कि पहले जो प्लांट में से सिर्फ एक्सट्रैक्ट करके पत्ते ही खाए जाते थे, उनका सूप ही बनाया जाता था, अब उसमें से you can actually extract different molecules, and then you can study the application of different molecules. Now I'm so far touching only the chemistry side and the analytical chemistry side. I'll come to the biology side. Study करने के लिए, what are you studying? You need to know what you're studying, right? I didn't do anything. So as these technologies, this knowledge became more and more available in different disciplines, different areas, applications started to become. Don't worry, I'm going slow. I'll jump quick, bring you to the currents. But I'm just trying to give you an appreciation of how it takes time for science to move. And then, as you see, now opioid, many parts of the world, opioid is a huge problem. Problem is because regulations के कारण, because it gives high and people are using it left and right, and without knowing उसके जो negative consequences, and that is why it is a problem. But it also has biological application. That's why it's a problem. Now, can that application be converted for a useful purposes, and then it becomes a medicine? So opioid-based medicines, again, morphine, right? Just Civil War के दौरान, it was one of the things which was more used, but then slowly regimented तरीके से it developed and became into a medicine. I'll give you a few more examples, and then I'll bring back quickly to what is the current, जो modern chemotherapy, what it is and where we are, and how that works. So quinine, malaria. This part of the world, we know the term malaria very well. Western part eradicated a long time ago, not. So malaria के लिए cinchona bark जो है, quinine जो है, quinine is basically a molecule which is extracted, from the bark of the cinchona tree, right? And that was observational, because if you take that was again purely observational, that if your cinchona bark जो tree है, उसके bark जो है, if you किसी को मलेरिया हुआ होगा, these are all human observations. किसी ने गलती से, क्योंकि फार्मिंग इज द मेन थिंग, वहाँ उठा के खा लिया कर लिया होगा, उसका बुखार ठीक हो गया होगा, there is something in there. So that observation led to actually identifying what is in that bark. That led to cinchona, right? That led to quinine. Now these were based initially only on the extraction, because extraction technology was available. Extract करके बना सकते हैं. But the fact is, to spread it more places, then organic chemistry, analytical chemistry became more and more accessible and more advanced at that time. कि उस cinchona bark, because bark is not one molecule, उसमें hundreds of molecules कुदरत ने डाले हैं. But how come you just get to quinine? So you extract it, analytical chemistry advanced होने लगेगी. You extract various techniques and you identify quinine is the molecule, which is the one which has the pharmaceutical application. And that is how that evolved. And then, as you can see, the cinchona bark in the 17th century, those observations were made in 17th century. So think for a second. I mean, I'll bring you back to, but again for the appreciation, think for a second, okay, we are in 2025 as per the modern calendar, and of course world existed even before that. Up until 17th century, कुछ ज़्यादा पता नहीं था. There wasn't much information known. So this is all past 2.5 to 3 centuries. और उसमें भी अगर देखें, I'll share with you examples of इस past 3-4 decades जो advancement हुई है. So on the human scale, to make an advancement, it takes a long time. Long time. So as you can see here, कि ये करते कराते यहाँ तक पहुँचते हुए भी ये सिर्फ 17th 18th सेंचुरी का है. गिव यू एन एग्जांपल ऑफ 1904. तो ये सोच के देखो कि 1904 पहले 1904 years, if you start with that, and prior to that there was no such information available. लोग क्या करते होंगे? Therefore the lifespan was also in 20s and 30s. Lifespan wasn't that much at during those times, because the disease, I mean, if you look at the history, if you read the history, famine, भूखा आया, जो कुछ भी हुआ उसके कारण इतनी diseases फैली, and then and the population got wiped because there was nothing to fight against. So our understanding of what we call the modern science, as you can see, is very recent. It's evolving and it's very recent, while this machinery is very complex. And so therefore, again, I'm connecting you back to just to give the appreciation of experimental therapeutics. Okay, techniques that has brought us to where we are today has taken a lot of hard work by the human as a race to bring us to where we are right now. So these are just just to see. And then the first synthesis. Now quinine extraction was known in 1747. Think for a second how the speed with which it moves. 1747 it was known that cinchona bar has a therapeutic effect, right? But the molecule to synthesize first synthesis was done in 1944. 200 years to make a molecule. So just imagine on the human scale, our understanding, our knowledge of each field, it takes a lot of, and 200 years to come up with the synthesis of that molecules. Uski application was known 2 centuries ago, nearly 2 centuries ago. So that's how, just to give you a few examples, the appreciation of the, what has taken, the historical background, what has taken, and how the science have evolved. Now again, this is, because many, as we call fields, most of you are chemists, all of you are mostly chemists in this, so we are talking about chemistry focused. But the fields, when we talk about, as I mentioned, biology, even biology you can subsect depending on किस तरह की biology आप कर रहे हैं, pharmacology, toxicology, cancer biology, chemical biology, in general I call it disease biology. Each one can be broken down. Immunology now, which is the field which is evolving so rapidly. All of these are highly complex and were not advanced. So this is just based on giving you appreciation for the chemistry. So these are all observational again. And then again, as human races, झगड़े होते हैं, wars होती हैं, तो दुश्मन के खिलाफ many things are used. So I'm showing you here example of one of the observations that was made during the war. So 1917, that's over a century ago, 108 years ago, right? Then troops in Belgium, they were fighting, they observed that there was some smoke coming out, and that with that smoke they had blisters all over their body कुछ घंटों बाद में, and pain, excruciating pain, and many of those people died, troops died. Then the question was, what was that smoke? Anyway, a lot of science, observation, analysis happened. It turned out that it was basically mustard gas which was used against them. And mustard gas, as we talk about chemical weapons, usme bhi mustard gas is used. So basically what it does is it attacks your body ke cellular functions and has all this toxic effect. So that observation was made in 1917 after that attack, that they see that the gas has something happening, which is causing them for all the blisters, their body not reacting, reacting to that gas and having very toxic effect. So the first use that happened, and there were 10,000 people who died because of that smoke. And that smoke basically was nothing, that mustard gas. Now when that happened, then obvious curiosity is, how does it react to the body? What does it do to the body that it caused such a severe reaction and 10,000 people died? So there was obviously curiosity, scientists, academic curiosity from them to see why it is that smoke was so toxic. So two scientists at the Yale University, Louis Goodman and Alfred Gilman, they were medical doctors, medical researchers. They were curious to see that what was that happened. And when that happened, what was the reaction? Body reaction to that mustard gas. And they were curious to see how do we measure the effect that happened. So what they, again, regulatory, you have to go to the government, you have to get the approval to get the samples of the troops, those who survived and those who are affected. So it took them a couple of decades or more to get those samples or to get the information about the other troops, those who are affected, and study that information. So what they found out was that, as I have noted here, that the number of immune cells in their blood was affected. It was affected surprisingly low, the white blood cells. And so when they started to think, if that happened, then maybe there could be a useful therapeutic application that can be developed with the mustard gas. And with that, they started doing experiments. That if that happens, then probably it could be used for some biological disease that we don't know of. And then they started to treat. They did many of the the lab studies, and they did the animal trials. They found the human volunteers after the successful animal trials to see that if they see some positive effect on the disease, and specifically they were studying leukemia and lymphoma, the cancers, the blood cancers. That's what they were researching on, and so based on the medical records that they observed, based on the observation that the immune cells specifically number goes down by the treatment, and since they were studying leukemia and lymphoma, they started to develop method to study by treating leukemia and lymphoma cells and saw the positive effect. They took that knowledge to start doing animal studies, study the same effect in animals affected by leukemia or lymphoma, and see that again they observed very positive outcome by the treatment. Then they found some human volunteers who voluntarily, they had the disease and it was not affecting, and then say, okay, fine, there is nothing else left, try this. And those human volunteers also. So the first time, the formal record of giving a known, well-studied, at that time well-studied, a candidate to a patient with cancer was, as you see on my screen, it was at 10 a.m. on 27th of August 1942 to protect the identity of the patient. It was called John Doe, no name given to that, was treated with that mustard gas. And that cancer patient started to improve, started to feel better, started to eat better. Okay, his condition started to improve. And unfortunately, he didn't survive after some time, but the reaction was that by using that mustard gas, by using the chemical that they converted into a drug, there was positive response by the cancer patient. That is the first known record of chemotherapy that you hear so frequently now. So just to give a historical background, so centuries and centuries, nothing known. 17th, 18th, 19th century, some observations. That observation leads to systematically developing some approaches, some strategy, and that is what led to the first known chemo treatment. So 1925, so that's what, 83 years ago, was the first chemotherapy was given. So think about human race, we are in 2025, it's only very recent कि actual chemotherapy जो है दी गई, and even though that we know that there is a lot more that needs to be done than has to be done in order to cure many of the diseases that we have. So anyway, because the analytical chemistry was known, known in the sense there were tools of analytical chemistry available to be able to identify the structure of a molecule. This is, as you look at the structure of mustard gas, it's a very simple molecule, but see how much effect it has on the human body. Blisters and such a toxic effect, but it's a very, very small molecule. So once that was known, so I have shared here a few more examples of similar molecules that were developed. So when that was there, people started obviously knowing that if this molecule have such a huge reaction, positive or negative, depending on the dose and how you treat and what you treat, then there is potential that similar molecules can be converted into drugs, therapeutic application, and of course, creative chemistry mind started to come up with similar structure. And if you go into literature, there are, and you will hear from Dr. Singh and Dr. Bhalla about the metal-based drugs that are being used and for various treatment. But this is how it evolved. Simple observation, and then again, I have seen that chlorambucil, one example, which was from Dr. Hadow, had shown that its application from UK. But see, cisplatin is platinum-based. This is one of the structure, and then you can make various variations like carboplatin. In 1972, it was patented. But again, as you see, the speed with which now people thinking of more molecules that could have biological application speed up, because people know how to make molecules, people know how to analyze molecules. Then there was started to develop testing methods for testing those molecules. So it became faster and faster to identify more molecules. And I've just represented a few examples, but if you go into literature, you'll find hundreds and hundreds of these molecules which have been around just simple mustard gas, have been designed and tested and for various different diseases. So just to give you an appreciation of how just observational, and still it's observation. Observation has to be, as a scientist, you have to be very observant. But as simply an observation-based medicine to now systematically developing medicine that could be. Now this is all the chemistry side of it. That you have chemicals which are again coming from natural resources. आप क्या खाते हैं, क्या पीते हैं, उसका reaction to the body. Based on that, it became formulation. कि कितना खाना, क्या मिलाना. You make those formulations. And then as the analytical chemistry side, as the organic chemistry side, as the techniques of synthesizing molecules, as the techniques of analyzing molecules became more and more prevalent, more and more easier, it started to speed up the process to be looking for now not just few molecules which are coming from natural products, but you can now simply design different molecules. You can synthesize different molecules and test for various applications. And that application, again, in this case, we are talking about drugs for the human diseases. But again, that's how, treating even the plants. जो है organic manure जो है, it is what? Manure, simple manure की जगह पे organic आप दे रहे हैं. जो आपके कीट पतंगे जो हैं, those, even for the plants, if the plants के कारण जो पत्ते खा जाते हैं हो जाते हैं क्या है, it's a disease for those, right? You started, but point being, as a chemist started to develop, thinking of how many different molecules that can be synthesized, they can be tested, and their useful application. Now, when we talk about drugs, we are talking about drugs, about human drugs, and that's what we'll be covering. But that's again, it's a drugs for the plants, for the animal. They are drugs. If you can, you give the pesticides, you spray them, and they are healthier. Those are actually drugs for those. So, as the field of chemistry, I'm focusing on chemistry, and probably what we could do is, if you want, take 5 minutes break, and then I'll cover the chemistry, the biology side of it. So, but before you go, if there's any question, but that's just to give an appreciation of how things evolve, so that we understand कि अगर drugs हैं तो क्या है, where they come from, and how many different disciplines have to be brought together in order to understand the disease, the causes of the disease, and then to develop something which is safer and effective. Okay, any question? Anything you want to ask? Anything you want to share? Yes, go ahead. I'm sorry? हैं, but unfortunately कम है reported, क्योंकि जो उसमें investment, focus, determination, dedication is needed. It, for whatever reason, came from various parts of the world, and many of those were outside of India. Most of those, unfortunately, were outside of India. It's not that, I mean, if we see, the thing is, if you look at the history anywhere, if there is knowledge, I'll give you an example. That knowledge has to be protected. That knowledge has to be nurtured. That knowledge has to be brought together to the world level in a way that is effective and useful for humanity at large. And that, whatever emphasis उस पे दिया गया नहीं दिया गया, observations have happened, no doubt, but उसको किस स्पीड से आगे लेके जाना, उसको आगे लेके जाने के लिए आपको जो डेडिकेशन चाहिए कि कुछ भी हो जाए. अब many of these discoveries, for example penicillin, right, उसने अपने आप को सबसे पहले दे दी थी, but you need that guts के, you have to have those कि I'm absolutely right, this will work, I'm so right that वो कहता है I will be the first patient to take this. वो guts भी तो चाहिए. I mean वो it's not as simple to answer, but yes, you're correct. Most of these, as we see, the modern has come out of places, various parts of the world which is away from here. It's being recorded, otherwise I can give a non-political answer. It takes a lot. So as you see here, there are many reasons, as you see here. So that's a good question. Forget about, political, not political. The part is, it's a multidisciplinary process. What I'm talking right now, science, and even in science, I'm talking only the chemistry. You take 5 minutes break, I'll come to biology. And that's only science. Then we will come to the regulatory side. Then we'll come to the geopolitical side. What all it takes to get a drug to the market. And there are many, many. Insan is a complex animal to begin with, So anyway, I'll talk after that. So yeah, please. What else? Yeah, go ahead. किसने कहा आपसे कि they create problem? You have inside sources? It's not that they intentionally created. These are the side effects. You see, these are the side effects of any medicine that is developed, because medicine, as you will see during, I'll talk about that, because the idea is, well, I'll come to that, but just think for a second. If a medicine is developed, it's developed based on what we know today. We as a scientific community, we as a humanity, as we know today that this is the cause. And we try to, with all sincerity, with all the resources, with all the knowledge we have, we try to combat that based on what we know to do that. That's our collective understanding. But do we really know exactly कि उसके अलावा वो कुछ नहीं करेगी? Think for a second. एक cell, I'll show you a picture I think in the next lecture or something. I'll show you a picture. Cell is a soup of thousands of proteins and many other things. If you develop a drug for a protein target, इसका मतलब ये नहीं कि ड्रग किसी और चीज को टच नहीं करेगा. That's why it's a very complex thing. You have to do a lot of toxicity studies, and the toxicity studies are to make sure that it is safe. ऐसा नहीं कि वो एक चीज का जिसको बचाएगा मैं दूसरे से मार डालेगा. So side effects are, the goal is always to minimize the side effect as best as possible, so that they are not overburdened, that the positive effect is nullified or it's become less relevant. So that's, I believe, universally, every scientist working in this area is the interest to come up with something which has positive value, positive effect, but not have negative effect to the extent कि उसका कोई use ही ना करे उसको. That's not the goal. Yeah, go ahead. जी. ये बनाने वाले की रचना बहुत मजेदार है. You want an answer now? I can give you that, but I'll come to that. Keep that question in mind, and I'll explain to you how आपको मालूम है ह्यूमन बॉडी जो आप खट्टा खा लीजिए, आप मीठा खा लीजिए, आप कड़वा खा लीजिए, आप जितना मर्जी तीखा खा लीजिए. ये तो मैं बड़े सिंपल सी चीज़ें बता रहा हूँ. आपने खाना है, जो चने आपने चबाने हैं, फिगरेटिवली स्पीकिंग और रियली स्पीकिंग, जितना कठोर खाना है उसको भी चबा पाना, ये सब कैसे होता है? Keep that in mind. When we come back, I'll explain to you. ये जो रचना है बहुत मज़ेदार है, बनाने वाले की रचना. So human body at different parts has different pH. जाते हैं वो उसको तोड़ फोड़ के कर देती है एकदम छोटे छोटे molecules में. Then it travels to different parts of the body to get to what gives you the energy. ATP, ADP, ये सारे नाम सुने ना आपने, कि एनर्जी में कैसे कन्वर्ट होता है, वो सब कुछ जो है कैसे ब्रेक डाउन होता है. वो ब्रेक डाउन करने के लिए किस जगह पे क्या पीएच होना चाहिए, उतना ही पीएच है. ये मशीनरी बहुत मज़ेदार है, बनाने वाले की रचना. I'll come to that. So that is how it works. So आपने तो बड़ा सिंपल क्वेश्चन पूछा कि हमने एक फॉर्मुलेशन लिया, ठीक है. फॉर्मुलेशन तो हमने एक्सट्रैक्ट करके सिंगल मॉलिक्यूल ले लिया. अगर पत्ता लेंगे तो कैसे होगा? हमको तो कुछ भी नहीं आता, जो बनाने वाला है उसने तो सब कुछ बनाया हुआ है अंदर. दैट पत्ता ब्रेक्स डाउन, दोज मॉलिक्यूल्स गेट सेपरेटेड. द मॉलिक्यूल विच आर ऑफ लेस यूज दे गेट फिल्टर्ड आउट. The molecule which is of use goes and affects on the side where it should be affecting and giving you the positive or negative effect. So ये जितनी भी हम इंडस्ट्रीज देख रहे हैं ना, रियली स्पीकिंग, जितनी इंडस्ट्रीज हम देख रहे हैं ना आज के फार्मेसी, हम एक्सट्रैक्शन तो एक चीज़ है, सिंथेसिस, एक्सट्रैक्शन, बायोलॉजिकल रिएक्शन, राइट, फिल्ट्रेशन, जो फार्माकोलॉजिकल. I'll come to, I have a whole lecture on that. I'll come to जो व्हाट इज़ फार्माकोलॉजी, ये तो हमने नाम दिए हैं बेस्ड ऑन व्हाट वी नो, एज वी आर लर्निंग वी आर गिविंग दिस नेम. Body does that, has been doing this for